This meeting is being recorded.
Hello again. We are going to continue on chapter 2. I'm sorry, molecules of life. And so let's go ahead and, as I mentioned before, there are 4 organic molecules. Your textbook talks about it. What is the definition of organic? Here it gives you this one right here. That gives you, oh, did I turn on? I'm sorry, let me make sure my pen is on. Okay, so the definition of organic is given to you guys right here. Organic molecules contain carbon and hydrogen and are associated with cells. No, okay, carbon and hydrogen, or I should add carbon to carbon, carbon and carbon. Okay, and I should say, oh, sorry, and carbon to carbon. So, for example, the molecule CO2, if you look at it, this molecule, you know, when you've heard of this molecule before, CO2, you exhale it when you have some food, and then the food breaks down, and then you exhale carbon dioxide. What happens? Carbon dioxide is not an organic molecule. Or even carbon monoxide, you know, comes from the exhaust pipe of cars. That is not even organic molecule. Why? Because there is no carbon to hydrogen or carbon to carbon molecule in here. So that's why these molecules are not organic molecule. What is organic molecule? Well, glucose molecule, the sugar you had this morning with your breakfast, or proteins or fats, they are all organic molecules, which we will get into them in a minute. Okay, before I move on, the four major molecules, I call them macromolecules, or I call them building blocks. You know, I call them building blocks. Okay, these building blocks are for our living organisms. All living organisms are carbohydrates, lipids, proteins, and nucleic acid, which this session, the second portion of this chapter 2, is going to elaborate on them. Oh, okay, so molecules of life, again, continuing. Dehydration reaction, a type of synthesis chemical reaction that removes water, linking. You should see that with some examples. But what happens if you have, for example, quickly, if you have a sugar molecule and a sugar molecule here, when you want to combine them together, what happens to give you this molecule right here? So they combine together. What comes out of the combination of these two is a water molecule, H2O. So that process is called dehydration, because you're removing a water molecule from combining these two molecules together. I hope I'm making some sense. If you have the opposite of dehydration is hydrolysis. If you have a two glucose molecules are attached together, disaccharide. And what happens with some enzymes or something, you want to break this, okay? If you want to break this, opposite of that, you have to give it a water molecule. If you give it a water molecule, then you can break them down to 2 separate sugar molecules. And you will see these later on with some examples, I hope. Okay, let me get some coffee. Please forgive me, I'm drinking coffee or something while I'm lecturing. Okay, here we go, perfect. So here they're saying subunits. These subunits could be amino acids, it could be sugar molecule, could be fatty acid. Well, you will see them, I hope. And then you combine these two together, you get water molecules. That's dehydration, which I gave it to you right there, the definition of dehydration. Right here, opposite of that, opposite of this, it is right here, it is the, let's go back, hydrolysis. Right here, when you add water molecule to a molecule, and then you know, you break them apart. Right here, you add water molecule right here, you break them apart, it's called hydrolysis. Hydro You guys, it means the water. Lysis, it means breakdown. Let's go back. Lysis, it means break down. So when you break down these molecules, it's called hydrolysis, of course, with the help of water. Okay. Then let's go ahead and the first building block that I'm going to talk about your textbook. The first one, your textbook is measured building blocks, or I call them macromolecules, building blocks, macromolecules. It means macro, it means huge. They're huge molecules, you will see them. Used as energy source in plants, can be structural, made up of, very, very important, make sure you know this, made up of carbon, hydrogen, oxygen. Carbohydrates, they do not have nitrogen. And while your text is giving you the ratio of the water, of hydrogen to oxygen is 2:1. So these carbohydrates, they have a lot of hydrogen, okay? So, and then you will see them. So the ratio is 2:1. Let's move on. The monosaccharides, mono, it means one, okay? And saccharide, it means sugar. So simple carbohydrates are monosaccharides, disaccharides, made up of single sugar molecules, has a carbon backbone of 3 to 7 carbons. This is a pentose, has 5 carbons. Yes, you will see that. That is glucose, the most common, and is used in immediate energy source for our body. Here's a glucose molecule. I guess your textbook did not mention it. No, it did not mention it. But before these molecules become, I would like to show you something, becomes a ring, but glucose molecules are just straight. The carbons, they did not show it in this diagram, that's okay. But they are straight, and then what happens in plants, they go ahead and make a ring out of them, okay? So that would be an example of a glucose molecule. For simplicity reasons, a lot of times they do not show all of the carbons and hydrogens in here. So somebody look at this, they know it's a glucose molecule. It has 6 carbon, 12 hydrogen, and 6 oxygen. If you want, we can count them, but you do it at home, please. Here is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. And it has 5, as I said, 6 carbon, 12 hydrogen. You can count the hydrogens, and of course 6 hydrogen, and right here is another hydrogen. In this diagram, sometimes some textbooks draw it for simplicity reasons. They do not draw the carbons right here, or in the, here even get it simpler, they did not draw any hydrogens or oxygen here, but they just want to make sure that you know one of the corners, this is a hexagon, it has 6 sides. Maybe I should write that word. Glucose is a hexagon molecule. It has 6 sides, and then if you would, 1, 2, 3, 4, 5, 6-sided molecule. Do we have molecules that are 5-sided? Pentagon, you've heard of pentagon in Washington, DC. Yes, we have pentose, but I'll mention it as a pentagon. Disaccharides, di, it means 2, right? You have it on your prefix, suffix, and saccharide, it means sugar. So disaccharides, they are a combination of 2 monosaccharides together. And of course, joined by the process of dehydration. You're removing 1 water molecule from both of them. 1 water molecule is removed. That is sucrose, table sugar. The sugar you put in is sucrose. The sugar, it is in your milk. Some of you are intolerant to it. It's lactose, okay? And you cannot take dairy products because of that sugar. Your body cannot break down. Intolerance is also cannot be break down to lactose. Here it is. So you have 2 glucose molecules combined together. The OH of this one with the hydrogen of that one. It gives you the water molecule right here. And then these 2 are combined together. This sugar and that sugar are combined together by the oxygen in between them. So the process is combining them together, and I talked about it, dehydration. breaking them down. Of course, if you want to break that molecule, you want to break this molecule down right here, you want to break it down, you've got to give it what? H2O. When you give it H2O, right here, you break it down, those two. So the process is called hydrolysis. When you give it water molecule to break it down, it's hydrolysis. Lysis, as I said, it means breakdown. So polysaccharides, when molecules, they have more than two, you have, there is a number, I used to say that in biology, 1, but I guess here we are going to get it. It's a long polymer of glucose subunits. So what happens, a polymer, poly means many, right? You guys know this, poly, it means many. So polymer, so this is a sugar molecule, sugar molecule, sugar molecule, sugar molecule, and they are combined together, of course, then this is a polymer, okay? So a monomer, polymer, subunits, in this case, sugar molecule is a subunit right here. That is starch. So polysaccharide, example of polysaccharides are starch, glycogen, cellulose, very common in Mother Earth, okay? On the surface of the Mother Earth, anywhere in the ocean, in the sea, on Earth. But anyhow, energy storage for plants, glycogen, energy storage for animals, and then cellulose, we cannot digest it, is the one that is found in plants. Here is the one in potato. What happens, the starch is, I should write it down, starch molecules can be straight or they can be branched. What I mean, so the sugar molecules, oops, sorry, right here, they are, oh, yeah, really, oh, sorry, go back. Oh, right, yes, okay, so go back, please. Right here, they are either straight, okay, I hope you know what I'm talking about, or they're branched. And you will see the glycogen in human is not. And then that one branches. Okay, so the glycogen in human, you will see it's all branch. It's not straight. You will not find that in right here. Here is a glycogen, the carbohydrates, complex carbohydrates in human, which is always branched. And that is in the liver, in your muscle cells, you find glycogen. Make sure you're familiar with these terms. Later on, glycogen. Later on during semester, we need these terms so you can understand digestive system, you can understand respiratory, everything else. Excuse me, I'm sorry. Okay, the cellulose is a different story. The cellulose is a fiber that we cannot digest. I'm not going to go to alpha, beta, and alpha and beta linkage. You're not taking bio 1, but cellulose is also a complex carbohydrate. Plants make them, and then what happens, we eat them in order to have a better breakdown. You remember, I think I should mention this, when we eat, when metabolism, we talked about, I said I will talk about it later on, but metabolism is when you eat mashed potato, it has straight, for example, some of the starch molecules, polysaccharides in there, they are straight. So what happens when we eat them in our intestine, they break down to single sugar molecules right here. And then these single sugar molecules in our liver being put together only and only and only branched. So we do not have straight sugar molecules in our cells, if you would. It's all branched. So that's what, you know, so I gave you the definition of metabolism, example of metabolism. Breaking it down in our digestive tract right here, break down the starch, and then being put together in our liver, and our muscles as glycogen, right here. Okay, let's move on. Explain differences between, so make sure you know, please study these things and close your notes and be able to answer these questions. The next one is lipids. So lipids, they do not dissolve in water, unlike carbohydrates. They do, not cellulose, but sugar, the sucrose can be dissolved in water, as you know. Lack hydrophilic polar groups, so diverse in functions and forms, found in the form of triglyceride, fat and oils, phospholipids, and steroids. Most of the fat we eat in our diet is triglyceride, and you will see it. So about 80–90% of the fat we eat in our diet are triglyceride. And we eat some steroids, and we eat some, cholesterol. I should not say steroids, and phospholipids. So triglyceride made up of 1 glycerol and 3 fatty acids. Make sure you know these things: function in energy storage, insulation, cushioning, and some 2 forms, fat and oils. Fats usually animal origin and solid at room temperature. Oils usually plant origin and liquid at room temperature. Here we go. As you can see, these are like carbon, you know these things: carbon, 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 covalent bond, covalent bond. I forgot to mention that with glucose molecules, covalent bond, covalent bond. So between the carbon and carbon is a covalent bond. So a lot of carbons. At one end, it's an acid group, and the other end is a methyl end. So it would be just like this. So you guys can see this. This is just carbon, 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 carbon. One end is acid end, and the other end is the methyl end. Maybe I should, here we go. The 2 ends are different. Yeah, this is a better example. One end is acid end, and the other end is a methyl end. I hope you guys can see this, okay? So they are different molecules. If they are like hockey players in old days, they are missing some hydrogen, they're missing some hydrogen right here, then it's called unsaturated fatty acid. This is a saturated fatty acid. It means all of the hydrogens are attached to carbon. This is unsaturated fatty acids. Remember, carbon must have 4 covalent bonds attached to it. Very important information you should know. I will ask that in your quizzes and exams. Hydrogen has only 1 covalent bond. Carbon has 4 covalent bonds. Hydrogen has 1. Carbon has 4. Nitrogen has 3. And oxygen has 2 covalent bonds. But anyhow, so if the molecule is losing some hydrogen, then the bond between them must be double covalent bond. And you studied that. We talked about it in case of hydrogen in the past. So that's what fatty acids are. Now, if you have a glycerol molecule right here, and the glycerol molecule attaches, this is a glycerol molecule, attaches to a fatty acid, they draw a short chain of fatty acids usually. They're a little bit longer than that. Another chain of fatty acid, another chain of fatty acids. So you have 1, 2, 3 fatty acids attached to 1 glycerol. So the name of the molecule is triglyceride. And what happens? What process? The process of dehydration. So the OH of this fatty acid from the acid end combines with the hydrogen of the glycerol, gives water. How many water molecules? 3 fatty acids attached to a glycerol, 3. Okay, I hope I'm making some sense. And then if you want to break this molecule, your cells of your body will do this for you. If you want to break it, you need 3 molecules of water, which is called hydrolysis. So this is fatty acid number 1, fatty acid number 2, fatty acid number 3, and of course, this is 1 glycerol, which has 3 carbons. Okay, so let's move on. And the way I show it in the classroom, I say, here are my 3 fingers. I hope you guys can see this. This is a glycerol attaches to 3 fatty acids. So this is a triglyceride. You need 3 water molecules to break it down. And then when you want to put your cells of your body, put them together, of course, you form 3 water molecules. Okay, triglycerides are hydrophobic, so it will clump together in body as fluid. Emulsifiers are molecules that surround triglycerides and disperse or emulsify them. That is during digestion, which I think we will talk about it when we get to digestive system a little bit more, but it's good that you guys know this. Waxes, one fatty acid attached to an alcohol prevent the loss of moisture from body surface, and that is in cerumen or earwax. All of those, you will see them on top of the arthropods, insects. Fatty acids, long chain of carbons. As I mentioned this, I think this is a better diagram. So this is a fatty acid. Remember this? All long chain of fatties. The one end is acid end. So COH is acid. And then the other end is a methyl end. Can be saturated or unsaturated. I showed you guys what they mean. There are a few, look at these numbers. You don't have to memorize these numbers. Most fatty acids have even number of carbons, There are a few ones that have an odd number of carbons, just extra information for your brain. And then here it goes, talks about the saturated and unsaturated. I'm not gonna go ahead and talk about it anymore. Unsaturated fatty acids, which is like here, they lost the hydrogen here, and saturated fatty acid, unsaturated trans fatty acid, it means the trans, they draw a cis, so this could be, they're not showing a cis here. Trans fatty acid in one hydrogen is on this side, one hydrogen on that side, they call it trans. If both hydrogens are on the same side in some organic molecules, it's called cis. Okay. But anyhow, let's move on. Fatty acid concluded, saturated fatty acid contribute to atherosclerosis, a disease, accumulation of fats in your blood vessels, and then trans fats are man-made and contribute to heart disease more than other types of fatty acids. Or unsaturated trans fats or unsaturated fatty acids, usually plants do not make them, but very rarely, but they could be in plants, but the missing hydrogens are on opposite. Cooking causes this formation of unsaturated fatty acid trans fat. In past, the recommendation for total amount of fat in a 2,000 calorie a day, we're supposed to eat a diet that has 2,000 calorie a day, and then the diet was 65 grams. And now the research indicates that this type of fat eaten is more important than the quantity. Well, yeah, it flops back and forth, but one thing I want to mention while we're here, 1 gram of fat, it gives you 9 kilocalorie, okay? 1 gram of carbohydrates, it gives you 4 kilocalorie. And then 1 gram of protein, it gives you 4 kilocalorie. And then 1 gram of alcohol, it gives you 7 kilocalorie. I would like you to know this. Okay. It might come up during semester, but yeah, I would like to know these things. Okay, when you read a food label like this, very important, guys, please spend a few seconds on this slide. I know you guys just like to go fast. And then go to your kitchen and look at the food labels a little bit and see what's going on. The total fat, saturated fat, we said that is supposed to be good. And then cholesterol, okay, 30 milligram, 10% of the daily value. If you eat 1 serving size, that's per serving size, 220 grams. Serving per container, there are 2, I don't know what food is this. It doesn't say it, understanding, okay. But anyhow, dietary fiber 0, and then sodium, boy, this is loaded with sodium. That's what most food, processed food are. They're loaded with sodium and fat. That's very unfortunate. I thought Michelle Obama made a deal with Walmart to reduce the amount of sugar, sodium, and fat. But I don't know what happened after she left. I hope you guys watch your diet. I'd like to take a few moments and mention, please watch your diet. I'm not saying just get the habit of reading the labels all the time. Yeah, you should maybe at the beginning, and after a while it becomes a routine for you, what foods to eat, what food not to eat. But remember, your body is your temple and you should respect it. By putting junk food in there, a lot of sodium, a lot of fat in there, oh boy, you get to age, I don't know, 40, 50, maybe even younger, Houston, we got a problem, okay. But anyhow, just pay attention to your diet. The next one is phospholipids. Structure, you know, a good thing is to take a nutrition class. That's another thing I highly recommend for you guys. We cannot spend much time in here on nutrition. I used to teach it, but not anymore. But anyhow, that has the story of its own. Phospholipids structure is similar to triglyceride, but one fatty acid is replaced by polar phosphate group, and you will see that. Have a polar hydrophilic head. It has a head and tail. So the head is made up of polar hydrophilic and non-polar hydrophobic tail. So this molecule is very unique. It has a head that is hydrophilic and a tail that is hydrophilic, and the tail that is hydrophobic. Are the primary components of plasma membrane. It means cell membrane. Every cell has a boundary, plasma membrane, cell membrane, and then form a binary. Hydrophilic heads face the water solution, and then I hope right here. So that's a phospholipid look like. These are the two fatty acids. Okay, this is the head, and these heads are facing water, which is either outside of the cell or inside of the cell. Outside of the cell is water, inside of the cell is water. So that's where there are two layers, if you would, of these, like Oreo cookies. I had some Oreo cookies. I didn't bring it for you guys, but that's okay. The inside of the cell is full of water. Outside of the cell is full of water. The head are facing the water because they are hydrophilic. They love water. And these tails, they do not like water. They are hydrophobic. Okay? So that's what the cell membrane is pretty much made up of. Steroids. Lipids made up of 4 fused carbon rings. Remember why we say steroids are fat? Well, you remember this one. So if I make one ring, and you will see a picture, another ring, and another ring, and another ring, it has 4 rings. Can you guys see my rings a little bit? It's out of, okay, you don't know what I'm talking about. Okay, here I made 3 of them for you. It's hard. So right here, so it has 3 rings, and each type different in functional group attached to each other. That is, cholesterol is an example of steroid, and then components of mineral and precursor to other steroid molecules. That's right. So the steroid molecules like testosterone, estrogen, they're all being made from cholesterol, and then you eat a diet that is full of cholesterol. Here they are. Imagine if I make all of these carbons. Remember, on corners, I talked about that in glucose molecules. On corners, they are right here, they are carbon. And I can make this straight. Okay, the rings can come straight like this. So that's why steroids are fat molecules. They belong to these macromolecules. Estrogen, progesterone, and cholesterol is made from it. Okay, so I think that's all we talked about. Your textbook talked about steroids. Let's go to protein, very important. Function, support enzymes, speed chemical reactions, and support your skin as keratin, transport molecules. So when you take, maybe throughout the semester, we go over these things a little bit more, but make sure you know these, especially chapter 2 for rest of semester. We need that information. Protein functions continue, defense, hormones, and motions, all of this. Proteins are made up of amino acids. So you remember the subunit of protein molecule is one amino acid here, 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 and they're combined together. That's a protein molecule, okay? So components of amino acids are carboxyl and R group, each amino acid different in R group, which I show you a picture right here. Make sure you know the components of amino acids. Amino acid, they have an R group, which is different from one amino acid to the next amino acid. Okay, they have an acid group. You remember I showed them to you in fatty acid. It could be hydrogen here. When you saw the fatty acid, hydrogen here. So it has acid group. They have central carbon. They call this a central carbon. Okay, and they have amino group. That's what they call them, amino acid, amino group, amino group, amino group. And so they all amino acids have, and of course a hydrogen group, hydrogen here. So look at the hydrogen, look at the hydrogen, look at the hydrogen. So all amino acids have these four in common: acid, hydrogen, central carbon, everything attached to the central carbon, amino group. What makes them different, and we only have 20 amino acids, 20 amino acids. That's all we have, 20 amino acids. In nature, there is about 120, but in human has only 20 amino acids. Okay, and of course, they all have their names: valine, glutamic acid, lysine, tryptophan, aspartic acid, cysteine. This one causes your hair to be curly, or if you have too much of it in your hair by genes, if you do not have much of it because it has sulfur group, it forms a disulfide bond. Okay, here all your textbook does not have this, but I thought I'd give it to you. You don't have to know it, but look, they all have the acid group, the amino group, central carbon, and hydrogen. And this is the R group. This R group is different than this R group. That R group is different than that R group. So this R group is different than that R group. This R group is different than that R group. They're all different, okay? So how many you have? 20 of them. And of course, there are two amino acids that they have sulfur right here: cysteine and methionine, okay? But anyhow. So protein bound, the polar covalent bond between two amino acids is a peptide bond. That's what, you know, if you have amino acid here, you attach it to amino acid here, the bond right here, the covalent bond between these two is called a peptide bond. Okay, this is amino acid, amino acid, amino acid, amino acid. Polypeptide, 3 or more amino acids linked together. Here is an example. Of course, when you want to put these two organic molecules together, amino acids, you form a water molecule. OH of the acid from the acid of this amino acid and the hydrogen from the amino group of this amino acids combined together, and here is your peptide bond. And of course, you give out a water molecule. You want to break that one? You need H2O. Okay, I hope I made some sense. And of course, the process is hydrolysis, going this way, the process going this way is hydrolysis, going that way, the process is dehydration. You know this by heart now. A protein's three-dimensional shape is closely linked to its function. Denaturation, okay, when you break down a protein molecule, for example, you have a protein molecule like this, and you break it down like this, become like that, then this molecule cannot go back to become like this anymore. And that's called denaturation. When you change the shape of a molecule, that's very, very important in biology. That's called denaturation. So caused in extreme heat or pH. And also stirring can change, they didn't mention here, mechanical stirring. it can change the shape, disrupts the protein's function. Okay, levels of protein, that's another thing I want to talk about. Primary, secondary, tertiary levels of structure. So let's go over that. Very important. I did cut off some papers for you guys. Here are amino acids right here. When they're combined together, you guys can see this. When you're combined together like this, by peptide bond, one end of the protein molecule is acid and the other one is amino group right here. One end always and one end. You can see that. So there's an acid end, amino end. So when the amino acids are combined together, just straight line, that's primary. Okay, so this is an example of a primary structure. Then secondary structure, there are two different types of secondary structure. One of them is pleated, which is called beta, right here, you guys. It's called beta sheet. They are like this, like accordion. Any of you are familiar with it? They're pleated, okay? So that is an example of, and they're all amino acids in here, they're attached together. When they're pleated, and of course, hydrogen bonds, you remember that? We talked about it, cause these pleated sheets. It's called beta sheet. When they are helix, they're twisted like this, okay? When they're twisted like this, that is alpha helix, okay? Either beta sheet or alpha helix. And of course, again, hydrogen bonds. This amino acid here is have attraction with that amino acid there. So hydrogen bonds cause these twisting or pleated. So these two are example of primary and a primary straight chain, nothing big deal, okay? And then of course you have secondary. Example of a primary would be your insulin, okay, in your body. Secondary, it would be in your protein, in your muscle cells. So yeah, protein structure continue. Tertiary, the 3D shape of entire protein determine the 3 bonds type, ionic, covalent, and a hydrogen bond. Quaternary is, do we have? Yeah, we do have a picture of it. So if you have a protein molecule which is tertiary, it would be, for example, you have some portion of this molecule, they're all combined together, is tertiary, is pleated, and the other one is, for example, twisted. So they're combined together, and that would be an example of a tertiary. Where do you find this? Your myoglobin, the protein molecule, myoglobin. Myoglobin is a protein molecule found in your muscle cells. And of course, here's a disulfide bond. I talked about it. There must be a cysteine here and a cysteine here. Is another disulfide bond, cysteine here and cysteine here. And of course, these are all alpha helix. These are pleated, or maybe they are primary. So it's a combination of things. Then they did not show you, yeah, okay, quaternary, if you have this protein molecule twisted right here and pleated, that's one. Just, you know, you can separate them like your hemoglobin. And there is another protein molecule, but they are connected together, okay? So they are two separate, like your hemoglobin, the one that is found in your blood. Hemoglobin, okay? Hemoglobin is an example of a quaternary structure. Myoglobin is an example of tertiary structures. So hemoglobin, you have 4, they call it subunits. Anyhow, okay, so let's move on. And then I think the last one is a nucleic acid. I hope I stayed within my half-an-hour time limit. I did not have my watch with me this on my lap. I didn't have it earlier. Polymer of nucleotides. So what is a nucleic acid and nucleotides? I will talk about them. Let's see what, you know, I can explain things better with a picture. Sorry about that. components of nucleotides, very important. You should know that. A nucleotide, components of it's a phosphate, a 5-carbon pentose sugar. Remember that? A 5-carbon sugar molecule, pentose. We talked about it. And a nitrogenous base, which is, I will talk about those a little bit. Functions, store information of genetics, contain instructions for activities of essential life, conducts chemical reactions. Yeah. Okay, here it is, structure of a nucleotide. You have the nitrogen containing base right here. You have the sugar right here and a phosphate right here. And then I hope this is a nucleotide. Nucleic acid concluded. This is a deoxyribose nucleic acid. I think I talked about that. That's what DNA stands for right here. Stored information on how copy itself specifies. We'll talk about this when we get to genetics chapter a little bit. And then RNA is ribonucleic acid and various types, many functions. That is, some are the components of coenzymes, which are regulated enzyme, and adenosine triphosphate. ATP is like dollar in your body. Without ATP in your body, you cannot be active. You cannot do anything. So very good diagram, the differences between DNA and RNA. Very good, they did it for you. This has deoxyribose, this has ribose. What it is, this deoxyribose is a ribose sugar, 5-sided ridging, which does not have one oxygen, one O2. This one has O2, okay? That's it, that's one of the difference. And then these are adenine, guanine, thymine, cytosine in DNA, but in RNA, you have adenine, which you have it in DNA. You have guanine, which you have it in DNA. You have uracil instead of thymine. Okay, so you don't have thymine in RNA. You have instead you have uracil, and of course cytosine you have them. So 3 of them found in both. 1 of them is not found in the other one. Okay, double-stranded, of course, like this, and single-stranded, like that. And is it helix? Twisted? That's what the word helix means. Is it twisted like this? Yes. And the other one is not twisted. So backbone of DNA and some forms of RNA is alternating phosphate sugar group. So DNA look like a ladder. So the legs of the ladder, these are the legs of the ladder. They have phosphate, sugar, phosphate, sugar, phosphate, sugar, same as this side. Phosphate, sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate. So right here are the sugar molecules, right in here. I hope you guys can see this. Right here are sugar molecules, and these are the nitrogenous bases. So the rungs of the ladder, the steps of the ladder are the nitrogenous bases, which I will get into it here in a minute. So the base nucleotides are commonly identified in their base since they are components. Okay, DNA is double-stranded right here. However, take this ladder and twist it. I showed you a twisted molecule, of course, amino acid. But if you twist it, okay, the DNA molecule, this ladder, you take and twist it. I do not have one, please forgive me. Then it is double helix, and they call it complementary bases. Adenine always bind with thymine, and cytosine always bind with guanine. Right here, that's what I was talking about. The ladder that is twisted, okay? And always, 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 adenine, it's scary in biology to use the term always. Adenine binds with thymine and guanine with cytosine. Easy to memorize it, guys. See, G and C, they are almost look alike. The letter, I'm talking about the alphabetical letter. So easy to memorize. These two go together, and of course, thymine and adenine, they do not, the alphabetical letters, they do not look alike. So they go together. But these two, G and C, if you memorize, there are only 4 letters. In computers, you have 0 and 1, 0 and 1. Here in biology, we have 4 letters: guanine, cytosine, adenine, thymine. That's it. And here is the legs of, you remember I talked about the legs of the ladder right here. So here is a phosphate sugar, which is attached to the nitrogenous base right here. Phosphate sugar, nitrogenous base, phosphate sugar. Of course, this is RNA because what? Because it has uracil. DNA does not have uracil. Okay, uracil, it is in RNA. And of course, this is DNA. Let's go. ATP, another very important molecule we need to talk about right here. I'll let you guys read those stuff, but let me talk about what is going on with ATP. ATP has a pentose sugar, and it has adenine, okay, and it has 3-phosphate sugar. So what happens when you have when one of these phosphate breaks down, it releases energy. It has 3-phosphate, but one of the phosphate breaks down, it releases energy right here. Let me see. Okay, so, and that's it. All right.